All right, well, I'd like to start by thanking the organizers for the invitation to speak today, and thank you all for coming out so early in the morning. I'm going to tell you about our development of a cellular therapy for age-related macular degeneration using human embryonic stem cells. And really, it comes down to three things, and that's what I'll focus on. First, cultivation, and then installation, and then preservation. And I have just one disclosure. I'm co-founder of a startup company, and you'll see how they're involved in this effort a little later. Well, the National Geographic actually had a nice article on blindness in the world in September, and I was a bit startled by the statistics they cited. 39 million people are blind, and another 246 million have reduced vision. So it's obviously an unmet medical need of huge proportions, which varies from country to country. And if we think about cell therapy as a way to treat ocular disease of various types, the eye actually has certain advantages over other tissues. There are advanced surgical methods to access the back of the eye, the retina, for example. There are good ways to look at the eye using non-invasive imaging like OCT. And there are good endpoint parameters, not only just looking at an eye chart, but using things like microperimetry to map where the parts of the eye are working and where they're not working. And another advantage is we think we'll only need small amounts of cells compared to some other target within the body. Well, I'm going to talk to you about age-related macular degeneration and just have a few background slides. When you go into the optometrist and they give you that bright flash, they're taking a picture of your retina. It's called a fundus photo. And the normal eye is shown over here. Let's see if I can, I guess I'll use the, it's not working. Okay. Shown here, if you have early signs of macular degeneration, you get what are called drusen, these little spots, which can then progress either to the wet form or the dry form. In the wet form, there's leakage of blood vessels, and in the you can see bleed sometimes. Fortunately, there's a good treatment for this using VEGF inhibitors. But the majority of patients in the US get the dry form, where you have something called geographic atrophy forming here. And unfortunately, there's no good therapy for that. I mean, imagine if you went into the doctor and you were looking at an eye chart and it looked like this. You lose your ability to see in the central part of the visual field, which is what you use for high acuity vision. So it's especially devastating for patients. And something like 3 out of 10 people over the age of 75 in the US have some symptom of this disease. So what causes it? Well, we don't know all of the information, but we think in most cases the cell type that's important is called the RPE, the retinal pigmented epithelium, which is right here in the back of the eye, behind the photoreceptors, the rods and cones. And the RPE provides crucial support for the photoreceptors. Every morning, for example, when light hits your eye, the RPE phagocytoses little bits of the outer segments of the rods and cones that are shed. And without that activity, you'll go blind. So it's a key sort of nursemaid for the rods and cones. It transports a lot of material from the blood vessels, which are back here, and supplies growth factors for rods and cones. So once you lose your RPE, pretty soon you're going to lose your photoreceptors and lose your visual acuity. So how do we engineer a cellular replacement to replace the damaged RPE and hopefully do it early enough so that you can preserve the photoreceptors that are already there? So that's the idea. Intervene as soon as you can and provide fresh RPE to prevent the loss of the photoreceptors in the disease. Well, I'm going to tell you about an effort we call the California Project to Cure Blindness. It was funded by the California Institute for Regenerative Medicine, or CIRM, and it's a multi-institutional collaboration that involves USC, UC Santa Barbara, University College London, Caltech, City of Hope, and the startup company Regenerative Patch Technologies. We're led by Mark Humayun at USC Keck School of Medicine, and myself and David Hinton are co-principal investigators in this effort. And our job at UC Santa Barbara was to figure out how can you make RPE? How can you make it in a GMP-compatible method that could allow cells to be used in a clinical trial? And I'm going to tell you a lot about that effort, but then I'll tell you about how we then move towards the clinic with this cell therapy. So the challenges. First, cultivation. How do you make the cells? How do you differentiate the stem cell into RPE? How do you expand it? How do you bank it? Then installation. How do you deliver the cells to the back of the eye? And some are trying suspensions of cells that are injected. Others are trying putting the cells on a scaffold and delivering a monolayer implant. And then finally, does it work? Will the cells survive that you transplant? And will you be able to preserve the photoreceptors? So let's go through each one of these 3 steps. Okay, first of all, cultivation. First question we approached was, well, what should we start with? And it was known that both IPS and ES can be differentiated into RPE. And you heard earlier in this conference from Kapil Bharti, who's working to make autologous IPS for treatment of this disease. Our effort started with the H9 cell line, and I'll tell you a little bit about why as we go through. But H9s, it turns out, are pretty good at making RPE. And I'm going to tell you about 2 methods for making RPE that we've developed. One is a directed differentiation where you throw lots of growth factors at the undifferentiated embryonic stem cells and force them to become RPE, and it's a little faster. The second is actually easier, and we call it the spontaneous continuous adherent culture differentiation. And it's very simple. You take a culture of undifferentiated HESC and you remove the FGF. And they start differentiating into everything under the sun. But given the right conditions, you can make them via sort of a default or spontaneous pathway become RPE, and then you can enrich for those. So first, this spontaneous method, and you can see it's longer. We're up to 170 days here. Let me talk about that. And the first people to report this back in 2004 was the group from Advanced Cell Technologies, ACT, with Irina Klimenskaya. And what they found was if they just take a 6-well plate and they're growing HESC and they remove the FGF, it overgrows and they get this gelatinous mass of cells, but every once in a while they would see a little patch of pigmented cells. So they picked these out and characterized them, and they looked like RPE. So we started collaborating early on with them, and then struck out on our own, and we asked, can we increase the frequency of differentiation, and can we make this process xeno-free? So to make a long story short, by screening various types of matrices, substrates, and growth medias and cell lines, we found that the H9 line will develop into these pigmented cells, in some cases maybe up to 30 to 40%. And by transferring those to a new plate and using selective growth conditions that favor the growth of the RPE, we can get nice homogeneous cultures where if we stain for an RP marker, we can see 99% of the cells expressing RPE markers. And very importantly, we don't see any undifferentiated HESC markers in these cultures. We've characterized these cells extensively, and I'm not going to go through all the data, but suffice it to say that they're similar but not identical to fetal human RPE. And we're fortunate in this effort to have fetal human RPE as sort of our gold standard. We can compare our cells to fetal human RPE in terms of gene expression. We've looked at proteins they express, and we've looked at RPE functions both in vitro and in vivo. And we can see both ESRP and iPSRP express genes similar to fetal human RPE and different than the undifferentiated cells. So you may be wondering, well, 99%, what's the other 1%? And would that represent a problem? And we've looked at the possible contaminating cells using, for example, quantitative PCR, where we spike in a known contaminant like undifferentiated cells, and then we look for REX1, for example, as a marker of undifferentiated HESC. And if you look on this side, those are several different preparations, and we don't see any expression. We do see some expression, though, of MAP2, and we think that 1% is probably some neural cells, and they may be RP precursors. RP comes from the anterior neural plate, but we can see see the expression of MAP2. We can see some occasionally neural phenotype, but they're not dividing. Okay, so this original protocol used Matrigel, and although that wasn't a showstopper for the FDA, we wanted to get rid of it. And working with a variety of people and collaborating with the company Corning, we tested their Synthemax substrates, which started out as a peptide embedded in the plastic with a sequence from vitronectin that contained the adhesive motif RGD. And they now make Synthemax 2 that they can coat the plate with this mixture. And Brittany Pennington in the lab showed that Synthemax 2 works just as well as Matrigel, and we can totally eliminate that animal product and grow the cells in a xeno-free condition. And again, getting these colonies and then enriching them. Okay, so that method works well, but it takes a long time, 170 days. So we next tried to go to this directed differentiation. And the idea here is, can we mimic the in vivo process where we add growth factors to make these undifferentiated cells go through neural induction and then make early eye field, then optic vesicle, optic cup, and mature RPE. And we went to the literature and looked at what's known from studies of mouse and frog, and Dave Buchholz in the lab came up with what we call a 14-day protocol, where we first do neural induction and then RP differentiation by manipulating the Wnt pathways, the TGF-beta pathway, and FGF and IGF. And these types of experiments are quite empirical, where you test different things, but we've used this quite a bit now, where we start by inhibiting Wnt and TGF-beta, and then in the end stimulating the TGF-beta pathway with activin A. And this was published, but if we look at the expression of OCT4, for example, it goes down rapidly with time, and markers of RPE come up. And in that 14-day period, we can find cells that are 80% positive for early RP markers. They're still quite immature, and it takes an additional 60 days to get the mature RPE phenotype. But it is much faster than the spontaneous method. Now, this has been improved recently by Lindsay Leach in the lab, where she showed that late-stage Wnt activation actually improves the differentiation. So you start by inhibiting Wnt, and then in the end of the protocol, stimulate Wnt with this small molecule agonist of the pathway, and you can get even better RPE from this. Another problem with RPE, if any of you have grown it, is that if you continue to passage differentiated RPE, they undergo an EMT, epithelial-mesenchymal transition, after 5 passages, and they start to look fibroblastic and decrease their expression of RPE markers. And Roxanne Crows in the lab asked, can we avoid the EMT in cultured RPE? And Roxanne, who's here at the meeting, found that if you use the ROCK inhibitor and use a rapid passage method, you can rapidly expand these cells up to 13 passages and still get this nice cobblestone monolayer of RPE that are very similar to early passage RPE in their gene expression. They'll actually go a little bit further, but then eventually they also start to look fibroblastic. But this is a great way to expand your cultures and get lots of RPE. Okay, how does the rapid method compare to the spontaneous, the slower method? Are they the same? And recently, Lindsay and Roxanne asked this question by making cells by both methods and then doing RNA-seq. And they, in this study, focused on iPS RPE using 5 integration-free lines from Jamie Thompson's lab. And what we found is that using the spontaneous method, only 3 of 5 would generate RPE. However, using the directed method, where you're kind of sitting on them with growth factors and forcing them to become RPE, 5 out of 5 generated RPE. And then doing RNA-seq, what we found was that they're very similar in both cases to fetal RPE, about 5% of the genes differentially expressed greater than 2-fold, and even closer to each other. The rapid versus the spontaneous, only 1% of genes are differentially expressed. So they both appear to make a bona fide RPE. Okay, now I want to go on to the second part, installation. We can make RPE. How are we going to deliver it to the back of the eye? And are we going to use a suspension or a monolayer? And here's where we want to deliver it, in the subretinal space between the photoreceptors and the RPE. Now, when we did some early experiments with iPS RPE and injected suspensions in a collaboration with Pete Coffey's lab in London, we found that the cells mostly would just form these clumps. Here they're stained with a human-specific antigen. And that phagocytosis that I told you about doesn't happen very often. You can actually detect that by staining for rhodopsin, and it labels the outer segments. And you can see here's one cell that's taken up a few little bits of red. But in general, most of these cells are not in the right orientation to carry out phagocytosis, and they rapidly disappeared and died. And so what's going on? Well, it's known from studies of cell biology that cells need an extracellular matrix to adhere to. And if you detach them from the ECM, they undergo what's called anoecus, a version of cellular homelessness where the integrin receptors are no longer bound. These cells are unhappy and can undergo cell cycle arrest and sometimes undergo cell death. So our solution to this problem has been to use a scaffold. And we're using a substance called parylene. It's a xylene polymer, biostable, doesn't break down. And we collaborated with the engineers at Caltech to engineer a parylene that has ultra-thin regions, so it has really good permeability properties, and we coat it with vitronectin so that we have matrix that these cells can adhere to. Then we grow the RPE for 30 days, so they polarize with an apical side with these apical microvilli and a basal side. And so the idea is to deliver them so this apical side is right there in contact with the photoreceptors. Now, why a polarized RPE instead of suspension? There are other reasons too. Studies from David Hinton's lab, for example, have shown polarized RPE express lots of growth factors like PEDF stained in green here. If they're not polarized in suspension, they don't express much PEDF. They're also more resistant to stress. And this parylene has already been approved for use in the eye. It's actually used commonly to coat things like stents or electrodes. And it's been used for over 30 years. It's USP Class 6. And shown here are some data from the group at Caltech showing the flux across the membrane, and it's similar in permeability to the normal Brooks membrane, the extracellular matrix that the RPE sit on. The other thing is it allows folding, and I'm going to show you how that's useful for delivery in a minute. We've compared cells on a substrate directly head-to-head with a suspension in a nude rat model and showed that when implanted as a monolayer, they show improved survival. Do they work? Well, the in vivo proof of concept model we use is the RCS rat. It's not really a great model for macular degeneration. There is no good animal model, but it's a good model for RPE dysfunction. The RPE are dysfunctional in this animal because they can't do the phagocytosis I told you about. They lack the MERTK receptor, and they're born with photoreceptors, but the photoreceptors degenerate. So the idea is to implant our patch in there at postnatal day 28 and then measure photoreceptor survival and phagocytosis and visual function over time. And this is a really difficult surgery because the rat eye is so small at postnatal day 28. But just asking, can it rescue photoreceptors? Here's the retina in a cross-section, and there's the implant blown up here. This nuclear layer is the photoreceptors, and you can see good protection, whereas away from the implant, you see this photoreceptor layer has degenerated to only one cell thick. And this is 2 months after transplantation. What about phagocytosis? Well, again, staining for rhodopsin, now we can see a lot of these phagosomes inside the transplanted RPE because they're sitting as a monolayer in the right orientation to carry out this task. And if we look at the native RCS retina, we don't see anything. And so we do see phagocytosis, and we've quantified this in both cases, actually, and shown significant effect. Okay, well, moving now towards the clinic, how do we make this thing? It's a manufacturing challenge, and we've transferred our protocol to the City of Hope and made these under GMP conditions. The implant is about 3.5 by 6.2 millimeters, a little smaller than a penny, and here's a picture of one seeded with about 100,000 cells. The batch manufacturing process starts with a master cell bank of H9. Those are differentiated and expanded using that spontaneous method, the slower method. It's simpler and more amenable to GMP. We cryopreserve an intermediate cell bank and then thaw those out and grow them on the Parylene for 30 days, and then they're transported at 37 degrees to the point of care. We carried out IND-enabling studies using those GMP-produced cells, looked at toxicity, tumorigenicity, biodistribution, and efficacy at absorption systems in San Diego under GLP conditions, and tested the efficacy and local toxicity in that RCS rat model, tumorigenicity in the nude rat model, and we also showed we could deliver this in a pig eye. The pig eye is similar in size to the human eye, and the surgeons worked with us to figure out how to deliver it to the back of the eye, and I'll show you that in a minute. But these studies put together allowed us to show activity and safety in animal models. We prepared an IND submission for the FDA. It was 3,300 pages long. I didn't read the whole thing, but a big team put this together, and it was cleared by the FDA. Very importantly, we haven't seen any tumors in any of the rats, pigs, or the humans that we've done in the clinical trial. For the tumorigenicity study, we take the whole human dose, put it in a nude rat eye, let that rat live its entire life, then slice and dice every tissue and do H&E staining and look for human tumors, and we found none. Okay, so this is the strategy for delivery, and this is an animation showing how we hope this will happen. And good, it's running. So it starts with removing the vitreous in the middle of the eye, standard vitrectomy, and then some solution is injected behind the retina to cause a retinal detachment. So here's the retina, and the RPE endogenous layer is down here. We then cut a little hole in the retina, and here's our implant. We've developed a special tool that can grab a hold of a handle and pull this inside a cannula, and it folds up kind of like a taco, which protects the cells as this cannula is then inserted into the eye through the hole in the retina, and then the membrane is extruded where it unfolds in the subretinal space. So now the apical side of these RPE on top here is facing the photoreceptors that they have to interact with. The retina is reattached by putting oil in the eye that pushes down on the retina. And then there's some laser to cauterize any blood vessels that might have been broken. Okay, so this was worked out in the pig, and we've moved now on to a clinical trial. 4 patients have been treated so far. The sponsor of the trial is the startup company, Regenerative Patch Technologies, and we're currently recruiting additional patients. The clinical trial sites are the Roski Eye Institute at USC and associated, or not actually, other ophthalmologists in the Southern California and Arizona regions that refer patients. All the surgeries are going on at USC by Dr. Amir Kashani, who's the principal investigator of the trial. So what we're planning to do is treat 20 patients in the phase 1/2A, starting with a cohort of 10 that are already legally blind, where their visual acuity is less than 2200. Then in the second cohort of 10, we're hoping to go to patients that are 20/80. And there, we think we may be able to make a real difference because the disease hasn't progressed very far. If you wait too long, the RPE is dead, and so are the photoreceptors. So if you're just putting in RPE, you don't expect to see much in those patients. But if you can intervene earlier where they still have photoreceptors, we hope to see an effect. Okay, so, and you may be aware there are a number of groups that are going after macular degeneration with this cell therapy approach. And I wanted to acknowledge some of the various groups that are going after the same thing. And there are a lot of different approaches. Some are using RPE suspension from HESC or IPS. Some are using other types of cells, cord blood mesenchymal stem cells in Janssen, for example, neural progenitors. And then other groups are going after the RPE monolayer approach that I told you about. And that includes IPS-RP in Japan, also NEI's group, and the London group, where they've implanted 2 patients so far, going after a wet AMD that doesn't respond to Lucentis. So a lot of folks think this is a good target because of the advantages of the eye that I told you about. So some of the skeptics in the back row may have looked at this when I started and said, the end of blindness, really? Are we ending blindness? So maybe they should have a question mark here. But I think we're making good progress. And we don't know which of these approaches will work. We don't know, maybe it'll be a combination of different approaches. But it's an exciting time in the field because we're now starting to get results from human trials. So some think that cell-based therapeutics may be the next pillar of medicine to go along with small molecules and biologics, and we're hopeful that we can contribute to that. So just to wrap up, I told you about cultivation, 2 different ways to make RPE a spontaneous method and a faster protocol, and how to expand them for up to 13 passages. I told you about installation, how we've developed a novel surgical method to deliver a monolayer on a scaffold made of parylene. And then preservation. I've shown evidence that we can not only see survival of the RPE cells, but we can see preservation of the photoreceptors that they support in animal models. So we've started the phase 1/2A trial, and that is currently underway. So finally, just want to acknowledge the team at the California Project Cure Blindness, and the people in my lab, and especially funding from CIRM, Santen, and the William K. Bowes Foundation. And really want to acknowledge especially the patients. They're the real heroes in this, and those that are participating in our clinical trial. Thanks for your attention.
We have time for a couple of questions. I have one.
Okay.
Okay. So you showed beautifully how comparable your substrate is to Brooks membrane. Have you looked at metabolic? I mean, you looked at flux in general, but have you looked at metabolic transport, oxygen?
No, we haven't looked at oxygen. Dean Bach at UCLA has looked at H9-derived RPE in terms of the retinoid cycle, and they do make the 11-cis retinal that's needed. We've looked at barrier function, and they can make tight junctions and make a nice barrier, but we haven't looked at oxygen.
Yeah, or the other metabolics. Okay.
With the size of the patch you're introducing, what proportion of the normal macular area does that correspond to? How much of the retina can you support?
Yeah, so it's 3 by 6 millimeters approximately, and that'll cover the whole macular region. And so in the patients that we've implanted so far, we detach the retina in the area of their geographic atrophy where they've lost RP and photoreceptors, and we try to cover that whole area. In some patients, though, we've covered part of it, and so one of the questions is, will the patients, when we reattach the retina, fixate right on the patch, which is where the normal fixation is, and that's what we're starting to see.
Okay, thank you very much.
Thank you.
Our last talk for the session is going to be given by Dr. Paul Sieving from the National Eye Institute, and he's going to speak to us about gene therapy for retinal degenerations.
I'd like to thank the organizers for the opportunity to speak today on the topic of gene therapy for retinal degenerations. The intent of this talk is to just give you an overview of where things stand, and I really want to emphasize the diseases themselves. We hear about retinal degenerations, but what exactly does that mean? Let's start with the eye. The eye is particularly well suited for gene therapy, as Dennis Clegg also indicated, for cell therapy. It's a small closed compartment which allows for targeted delivery of the genes. It takes only a small quantity of the vector to do the treatment, and consequently there is low risk of systemic toxicity from this. There are other features, including the fact that there are a number of animal models that are homologous, genetically homologous, and one can explore the treatment benefits by simply looking into the eye, because the eye is optically transparent. Now, we're interested in getting to the retina at the back of the eye, but in addition to the back of the eye, we want to get the photoreceptors, which are at the back of the eye. They're on the bottom of the retina. Here is shown at the top. 3 different ways of looking at it: a cartoon, 2 cartoons, and in the middle, tissue section. The 2 tissues involved are the photoreceptors, rod and cone photoreceptors, and then the adjacent retinal pigment epithelium. So we have the same targets as Dennis has for cell therapies. Let's start right at the beginning. And the beginning in this case is the success of gene therapy for RPE65. That is a condition called Leber congenital amaurosis, identified some 150 years ago for children who were born essentially blind. The clinical designation also extends to children who have severe vision limitation and go on to loss of vision. Usually there's no family history, and there's a giveaway sign in infants and young children that the eyes are bobbing or there is nystagmus, and there is no electrical activity. The electroretinogram responses are blank. This is quite a rapid success story, going from gene cloning in 1993 to human gene therapy in 2008. Shown here is Al McGuire, part of the team of Gene Bennett and Al, who pioneered this, along with others whom I'll mention in a moment. Way stations went through animals. A mouse knockout, RP65 knockout, was engineered in the laboratory. And a naturally occurring animal, a Briard dog, was also identified with this trait. Now, the dog turned out to be important because the mouse eye is pretty tiny, and to manipulate that is difficult, whereas the dog eye is substantial in size, and the dog gives you a better readout. It wags its tail when it sees you. If the mouse wags its tail at you, I'd run away. Now, Al and Gene, certainly they are pioneers of this, but in fact there were others who were doing this quite simultaneously, down to the month at which they initiated the trial. Bill Hausworth, Sam Jacobson is the second team, and Robin Ulley and Robin Bainbridge. So there were 3 different trials going on concurrently, 3 different vectors, 2 different promoters, but the outcomes were safety, ocular safety, systemic safety. And along the way, was there any improvement in visual function? Indeed, there was. And the eye is particularly suited for asking that question because we have a number of measures. Acuity, can you see in the dark? Peripheral vision, peripheral fields. Functional mobility, if you can't see, you're not mobile. If you regain a bit of function, you can get around better. No toxicities were observed. Immune suppression was used in some and not others. And there is consensus on the bottom line outcome, which was there was benefit. In fact, it is agreed that there was substantial but imperfect recovery of rod and cone photoreceptor function. And this RP65 therapy is going to registration soon, we hope. Now, the eye is particularly vulnerable to genetic defects, but today let's talk about 2 forms of retinal degeneration. Stargardt maculopathy. The macula is the center of vision. You're looking at the slides with your macula. If you lose your macula, perhaps because of Stargardt or other conditions, you will not see the slides clearly. Or a second form where you retain the center vision, retinitis pigmentosa, at least until the end stage, but you lose your peripheral vision. So those are 2 principal types of disease that one would want to treat. Let's look at that retinitis pigmentosa again. This was a patient of mine when I was faculty at the University of Michigan a number of years ago, a 73-year-old woman who had a rhodopsin mutation. And the fundus photograph at the edges shows the visual pigment of degeneration. But in the center, you can appreciate a little better retina coloration. And that translated into a central tunnel of vision, whereas she has lost her peripheral vision. She could technically still drive a car with her acuity, although not in my neighborhood, please, because you can't see the kids coming at you. In fact, her mobility was somewhat limited by this. She died, bequeathed her eyes. We had those eyes within an hour of death. And this is the eye cup, and obviously it looks like the fundus photograph. And looking at the histology across the macula, one sees very few cone photoreceptors remaining compared to a normal eye. And those normal cone photoreceptors have long inner segments, a lot of mitochondria, and long outer segments, which have the light-sensitive opsin, whereas her outer segments are minuscule. And this is an eye with 20/60 acuity. I'm not showing you the periphery, but it was just scar tissue. Now, question you've got to consider is this still something you can rescue? Conventional wisdom, clinical wisdom, says this really is too late. This is on a pathway to loss of cells and blindness. But maybe we could slow it down, and that would be a laudable goal. Now, the photoreceptor cell itself has many, many monogenic traits. This is just a short list of a few of them. And all of these are potential targets for gene therapy, and a number of trials, in fact, are going on for some of those traits. This is a fairly complete list, but I'm sure that I have missed some efforts going on. But at the top, RPE65, and then a type of retinitis pigmentosa, or the third one, RPGR, is RP3, an X chromosome condition. Or at the bottom, retinoschisis. I'm going to show you a few slides of that at the end. That happens to be, in part, a synaptic disease. It's one that I'm doing at NIH. Why pick these disease targets? Well, I would say these have been picked because we have some understanding of the function, the cellular dysfunction, in fact, caused by a genetic failure. And knowing something about the dysfunction, one can get a sense that in repairing the function, you might guess what the outcome would be. One of the important things to think about is whether you will get acute improvement or will slow down the rate at which natural history is causing progressive blindness. RP65 is at the top. The second one, choroideremia, is an interesting condition that is in very active pursuit. Here you can see there is just a little bit of retinal tissue remaining. The peripheral retina is gone. This person has severe tunnel vision. And so the treatment has to be directed toward this little tuft of tissue. That's a hard target. Rob McLaren is working actively on this condition, using an AAV2 vector to replace the REP1 gene or substitute. And he is reporting recently that there is some improvement, some gain of acuity in some of these individuals. Now, let's think then about how one does this. And there are some current challenges. AAV is the currently best vector. You've got to get it to the retina. The immune system pays attention to that. You can ask, how long does the treatment last? And are there other ways of doing it not currently being used? Well, let's just jump right in. Some genes are too big for AAV, and so lenti has been employed. ABCA4 is a large gene that causes Stargardt macular degeneration. I showed you that picture several slides ago. And a trial is actively underway using a lentivirus delivery developed by Oxford Biomedica. That same vector is being used for Usher syndrome. This is a particularly unfortunate condition in which these kids are progressively more deaf than they are born until they are totally deaf, and they become totally blind. It's a deaf blindness. And in both cases, the cDNA is too large to fit an AAV. But AAV is the vector of choice at the moment. There are a number of serotypes. Shown here are 9 serotypes. There are more serotypes. These target various cells in the retina, but there are some cells for which the vectors are fairly ineffective, and those would include the horizontal cells, bipolar cells, and amacrine cells, the cells in the middle of the retina. Ganglion cells, if you lose those from glaucoma, you might want to put in a gene to have neuroprotection. And among the current vectors getting to those photoreceptors, the best currently would be AAV9, which in non-human primate is particularly suited to the cone. So how do you get it to the retina? Well, you put it in the subretinal space. Again, Dennis Clegg already mentioned that. It's between the pigment epithelium and the photoreceptors. And you stick a needle through the eye, the wall of the eye, and make a little pocket of fluid with the vector in the subretinal space. Now, technically that's difficult, but doable, very doable. That's the delivery of choice at the moment. But it has a second problem, that you have limited space or limited bubble of fluid here, and so limited area that's treated. But it does have the benefit of being relatively immune protected. Now, a perhaps better way to do that would be simply to put the vector into the cavity of the eye, the vitreous cavity, make it an intravitreal injection. Very simple to do. It's an office procedure. Safe and easy. It gets theoretically to the entire retina. But that vector is more exposed to the immune system with consequent ocular inflammation. There's another problem, considerable problem. To get from the cavity into the retina means crossing the barrier at the retina surface, the internal limiting membrane, ILM. And that's pretty impenetrable to viruses. We hope so, because viruses have evolved and the eye has evolved over many, many, many millennia. And this is a protection for the retina. Well, Luc Vanderberg and Jason Commander a few years ago said, well, just peel it away. And yes, in fact, you can do that. You do a surgical vitrectomy, then you peel the ILM, in this case colored green, right on the surface of the retina, and you puddle on your vector virus solution. It's doable, and it does work. Here on the rightmost side, you can see that it works in a non-human primate. You can see the region that was peeled away across the macula. And there is better retinal entry of that AAV2 vector at 1 10 to the 11 virus particles. The same dose, just more generally into the vitreous cavity, doesn't get you very much. But both of those are less than a 10-fold lower dose put right into the subretinal space. So there is certainly room for development of better vectors. I'm going to show you two current strategies. You can know enough about the virus to figure out what is impeding its entry into the retina. That's a tack taken by Bill Hausworth and colleagues, including Shannon Boy in Florida. Mutagenize the tyrosines, threonines, and lysine residues at the surface of the capsid, and it works. The wild-type AAV is not getting in, whereas a 5 amino acid substitution certainly gains entry into the retina after putting it in the vitreous. And wild-type AAV2, AAV8, and AAV9, all three of those are shown here, work that Shannon did to increase penetrance into the retina. Just remember, you still have to deal with this host immune response. So this is one strategy. Another strategy, pioneered by John Flannery, David Schaefer in their two laboratories, and this work conducted by Denise Dalcar and Leah Byrne, using, I think it's a novel idea. You start with libraries that have many different mutant capsids. You put this, try a different pointer. You put these libraries into the vitreous cavity, wait a few weeks, and then take out the retina. And you figure out which of those viruses, in fact, are in the retina. They penetrated that ILM. Then you recycle them and iteratively enrich for AAV variants that reach the outer retina. And it works. In fact, one in particular, the 7M8 serotype of AAV2, works very nicely in mouse, shown here for retinoschisis XLRS mouse. The wild type control is not expressing protein. The wild type is expressing the RS protein down at the photoreceptors, and an XLRS mouse treated with a AAV2 7M8 vector in the vitreous that certainly gets into the retina and expresses. So this is a pseudo way to go. The problem with this particular vector is it was worked out iteratively in mouse retina. Doesn't work particularly well for non-human primate. But this group now, and others, now have repeated this in non-human primate and have some vectors that do work well. But there's a lot of, there's a ways to go on vector development. What about that immune response? I threw out a number of E10 and E11 virus particles. Just think about putting 100 billion virus particles into your eye. Well, you can get away with it sometimes, courtesy of observations that Peter Medawar made in 1948 when he put skin grafts into the anterior chamber of the eye and found that they survived longer than elsewhere in the body. And the eye became known as an immune-privileged site, along with some other organs of the body. That only goes so far, however, because it's really only a semi-protected ocular space. The best protected is that subretinal space. It's hard to get to and limited treatment area. When you put this into the vitreous, there is greater exposure and greater ocular inflammation. I can attest to that in the trial that I'm doing. But to date, AAV is the generally safe and well tolerated, and it does manage to deliver sufficient gene to have therapeutic benefit in some cases. And then we get to this persistent question of, does it work, does it last? How long does it last? This is a landmark paper, 2013. I'd recommend you read it because it's a very nuanced paper. But let me just give you an orientation for reading that paper. The top panel has the vector put into subretinal space, so this is a part of the retina that was treated, and the red part is a comparable region right next door that was not treated, did not receive the vector. The middle panel is function, and the lower panel is structure by optical coherence tomography. And you can see that the treated retina at time 0, when the vector was applied, suddenly gained sensitivity, gained tremendous amounts of sensitivity, log units of sensitivity. But the structure continued to be lost at the same pace as the control region. It's a very interesting paper because in addition to these human data, these are all treated patients, RPE65. In addition to those treated patients, there are some dogs that were treated and at various ages. And the conclusion of this group Sam Jacobson, Art Sidesian are consummate clinicians. Their consideration says that once degeneration has happened, it may be too late. So think back to that 73-year-old RP patient I showed you that had already lost many cells in the macula. That just may be too late. Here's another durability study. Same RP 65 patients, done 2 years later, same group. The red area, the reddest area, shows the area of vision is expanding across the first 2 to 3 years, and then it shrinks. Or you can plot it formally in a graph, and first the area expands after the vector is deposited, and then it constricts. So that functional improvement was not sustained over the 6-year period of time. But before you throw in the towel, let's just think about some of the ways that that could be happening. It could be an irretrievable gross failure, or it could be that the dose was too low. Perhaps a higher dose would better protect those rod photoreceptors. Or perhaps those eyes were treated too late and you've got to treat at younger ages. So there are ways that one can think about improving durability, and I personally think this will be successful. But it is something that awaits future data to figure out. Well, let's talk for a moment then about gene editing. The world is full of CRISPR-Cas9, as we know. This is one of the first off the block, done by Steve Tsang at Columbia, published last year, in a mouse with PDE6 beta mutation, very rapid death of photoreceptors. He corrected, using CRISPR-Cas9 techniques, a mutation in that gene. And whereas the mutant animal here shows that depigmentation of the RPE, kind of like the choroideremia patient, if you remember, the health of the RPE is better restored in this repaired retina. And function goes from the flat red line on ERG in the mutant to substantial retention of function in the treated animal. So it's possible to do this in vivo. A very interesting paper was published just recently, 2017, by Zhuzhen Wu, happens to be at the National Eye Institute. But it's a very clever strategy, and that is ablating, getting rid of a gene, the NRL gene. NRL stands for neural retinal leucine zipper transcription factor. I'll ask you in 3 hours if you remember that. And it is known to be important in retinal cell development. But it turns out also to be critical in maintaining homeostasis of mature rod photoreceptors. And when you ablate the NRL in rod photoreceptors, their phenotype migrates to have characteristics of cone photoreceptors. And the mutation, PDE6 beta, for instance, is going to affect the rods and not the cones. So by causing these rods to have cone characteristics, turns out they become relatively protected. Or, again, that 73-year-old woman who has a rhodopsin mutation. Rhodopsin is in rods. If you cause the rods to have cone characteristics, you would protect that woman, relatively protect her from the rhodopsin mutation. And in fact, Zhuzhen and his group have shown that here in PDE6 beta mouse, which has lost most of the rod photoreceptors. There should be 10 rows of nuclei, there's 1. But knocking down NRL at a young age protects the rods. There's a second effect, and that is that when the rods die, the cones die. You and I have 100 million rods in our retinas, each eye. We have 6 million cones. If you kill 100 million rods, the 6 million cones don't last forever. And so this animal had protection not only of rods directly, but cones indirectly. And it worked for a rhodopsin knockout also and for a rhodopsin mutation. Very clever strategy. Maybe it's more generalizable than a direct mutation, handling a direct mutation. Well, there are other topics. We need new models. Maybe CRISPR could be used to generate some primate models that would really be analogous to human disease. I want to end this with 4, 5 slides of one more disease, and that is something that I'm personally working on for a number of years, X-linked retinoschisis. This is a recessive disease. The carrier females pass it, the mothers pass it to kids, sons who are affected. It has both retinal structure problems. Here you can see the cysts in the retina, or on the fundus photograph, the radiating pattern of intraretinal cysts. Schisis is a German word for cysts, delamination, splitting. It has structural effects and it has functional effects. I've got to explain this because in a moment this will turn out to be important. The normal has an A wave and a B wave on the electroretinogram. The A wave is photoreceptors. The B wave is the adjacent neuron, the bipolar cell. You only get from photoreceptors to bipolar cells through a synapse. In the retinoschisis animal, the photoreceptor A wave is great, wonderful. What happened to the B wave? That's evidence of a functional problem. So let's look at that synapse. But first, in the wild-type retina, the retinoschisin, shown here in red, is prominent in the rods, inner segments. We're trying to figure out why it's there. We've looked very hard, can't quite get a full clue on that. It's also prominent in the rod synapse onto bipolar cells. These are the bipolar cell dendrites with dendritic tips, and the bipolar cell itself is at the bottom of the slide. Now, the dendritic tips with the synapses are shown here, these little tiny individual nubbins. And in the untreated eye, these signaling molecules, important for the synapse, are delocalized. They're streaming right down the dendritic shaft. But after we treated at 4 weeks of age and looked 2 months later, the molecules relocalize and the synapse begins to work. Here's that ERG again. Photoreceptor, untreated in black, the B wave is missing. But after you repair that synapse, the B wave comes back, the green line. And this the green is from the treated eye with cavities closed compared to the untreated fellow eye. So right here you see 2 possible landmarks for outcomes of a human clinical trial. We have been following a large number of these patients at NIH. And at young age, here 20 years old, the retina is thick because of the cavities. It should be this thick, about 250 microns thick. Here's one that's 600 microns thick because of the cavities. And the thick retina holds till about age 45 or 50, and then after that the cavities collapse and it's probably too late to treat. Vision does not come back here, but vision is lost because photoreceptors are lost. So we have a study going on. Conventional idea, safety and tolerability, that's where you start, phase 1. We're using an AAV8 vector, putting it into the vitreous. That's an entirely different story why we're putting it there. It's a dose escalation study. And at the higher doses, like everyone, we are seeing some inflammation, and we're working through how to deal with that. And it's getting very interesting. So let me then conclude. There certainly are a lot of human diseases that cause retinal degeneration, and there's a lot of opportunity. There are some really good animal models, principally mouse and dog. There are about 20 or so dog models, direct gene homologies to human. But further development is needed. Different capsids, better vectors, need to deal with the immune response. How do you make this a longer lasting treatment? And how do you deliver it into this vitreous cavity and get it into the retina? There's a prize there. But I would say that substantial progress is being made, and I think it's really an exciting time for gene therapy. So thank you.
We can take a couple of questions.
Thank you very much. Wonderful presentation. In your retinoschisis preclinical studies, how did self-complementary AAV8 do in the vitreous of a large eye?
Excuse, I didn't hear that. How does the
SC AAV8 perform after an intravitreal injection in a large eye? Because I know you presented the issues with AAV2, so I'm wondering about this difference.
How does AAV8 perform in a large eye?
Yeah, like efficiency of targeting the RGCs or...
Oh, I see. AAV8 is good to get to the outer retina. It's good for particularly photoreceptors, and that's one of the targets that we have here. The RS protein is a secreted protein, and there's certainly a lot of it in the photoreceptors. If we can get them to express this, I expect that it's going to spill over to the adjacent bipolar cells.
So you find with the intravitreal injection of AAV8, you have enough expression?
Yes, right. Well, as I said, we can talk about that offline, but in the mouse it works very well. We put it into the vitreous of the mouse, and it immediately targets to the photoreceptors with good expression there. And the question, among others, will be whether that is going to extend to the human retina also. It's an open question.
Could you say a little more about the inner limiting membrane and why it's such a tough barrier, what its composition?
There are a couple of molecules that bind viruses and hold them up at the surface of the membrane. It is in part the membrane that is inhibiting virus from getting from the vitreous cavity to the retina, as shown by that peeling technique, peel and puddle. There are some inconsistencies with that idea in that if you put AAV2 into a wild-type eye vitreous, it does get to the ganglion cells, which lie at the surface of the retina, but underneath the internal limiting membrane. It's a bit of a confusing story. The observation has been made in a number of laboratories, including ours, that ILM appears to be a barrier. It's an imperfect barrier, but it's sufficiently impeding vector flow that if one can figure out a way to get the vector through it, you could probably use 10-fold less vector, which would considerably change the immune response profile. Yes?
Could you comment a bit more on the immune response in the eye? Are these antibodies diffusing into the eye against AAV, or is it local innate immune response?
That's an inside question, isn't it? There is a difference of opinion on whether the neutralizing antibodies are in the vitreous already, or whether they are in less amount in the eye than in systemic circulation. It does appear, at least in some hands, that there is resident neutralizing antibody to these vectors within the eye. It takes a number of days after administering the vector before one gets a frank inflammatory response. My lab and other labs are trying to look at the nature of that response, whether it's T-cell mediated, which we and others think it probably is. But back to your question, there is the conventional idea at the moment is that if a patient has neutralizing antibody titer of 50 or more, some quite low number, those antibodies will immediately disable the vector when it is put in site. It's an area that needs exploring.
Thank you very much for that wonderful talk. Thank you, everybody.